Thank you very much, Mrs. Egi Arguni, for leading the session, and thank you to all speakers. Well, ladies and gentlemen, now we move to the Symposium 8 under the topic of Monitoring and Impact Study. And this session will be led by our moderator, Professor Damayanti Buchori from IPB University. She is the professor at the Department of Plant Protection, Faculty of Agriculture, IPB University. Professor Damayanti is an insect ecologist by training with a passion for landscape ecology and land use change. She completed her undergraduate degree at IPB University, Department of Plant, Pets and Disease, Faculty of Agriculture. Following this, she completed her master's in entomology at the University of Illinois at Urbana-Champaign, USA. She continued her doctoral education at Ecology, Indiana University, Bloomington, USA. Currently, she is a professor, and she is also the head of the Center for Transdisciplinary and Sustainability Science at IPB University, Indonesia. She is also the lead co-chair of Task Force for Food Security and Sustainable Agriculture, T20 2020 Indonesia, one of the engagement groups of G20. Well, ladies and gentlemen, please welcome Professor Damayanti Buchori to lead the session.
Okay, thank you so much. Hello, everyone. My name is Dami. Damayanti is too long, so it's Dami for short. And I would like to thank the organizer for inviting me to become the moderator for this session. So our session today is Monitoring and the Impact Study. And we have three speakers today. The first speaker is Dr. Warsito Tantowijoyo. The second speaker is Dr. Riris Andono Ahmad from UGM. Pak Warsito is from WMP Yogyakarta. And then the last one is Dr. Kat Edenborough from, I think from a little bit of information that all the speakers will have 20 minutes, and we have two live speakers. The last one will be a recording, and then it will be followed by a question-answer session. So all the questions, please, you can write down on the chat box or on the Q&A, and then I will read it to you later when the time comes. So without further ado, let us just start with the first speaker. I think there is a little bit information that I can read for the first speaker. Yes, here he is. Dr. Warsito Tantowijoyo. He is lead an entomologist and also team leader at the WMP Yogyakarta and received his master from Bogor Agricultural University and received his PhD from the Center for Environmental Stress and Adaptation Research from University of Melbourne, yes, from the Faculty of Science. Dr. Warsito, I've known for so long. He was one of my most bright student when he was an undergrad and also a master. So I'm very pleased to moderate this session for him. So Dr. Warsito, you have 20 minutes.
Thank you, Budami. Thank you, organizer. Thank you for audience for all. Good afternoon. And thank you also for Ari and Budami as my guru, my lecture. So can you hear my voice?
Yes.
And then can you see my slide as well?
Yes, if you can. Yes, perfect.
Yeah. Okay, thank you. So probably a little bit away probably from the topic, but I try to make it not really redundant with the previous presentation. So it's mostly about entomology, looking for the status and what actually we need. Just right, because my boy coming to the room. Sorry, this is the problem. Sorry. Oh, sorry, I'm back. Okay. Then I will present about status of Slovakia. but then actually more focusing on more not really explicitly talking about monitoring, but I will highlight what actually we monitor during the Wolbachia establishment in Yogyakarta. So this is just about the overview. I think it's Katie and also Ari and also Buut already mentioned that we started the project actually in the 2011. And then this is the last year of the project. And we look at how is actually the story of the Wolbachia deployment. We start actually 2011, but we success to release actually 2014, because before that we're dealing with the regulation, we're dealing with community preparation on the lab, etc. We start from very small area, 2 kilometer square, with the population is about 2,000. But this is the most complex thing, because the first is usually the most difficult things. But we success with this kind of the process, and I will mention later. And then we getting to the bigger, because the first is actually talking only on the entomology, and then second is about the impact of study. We're talking about the Wolbachia, mosquito, and dengue. So we wide the area of the release to the entire of the city of Yogyakarta. The size is about 32 kilometers square, with the population is 500,000. But this is not really, and then we got a success there, and when we jumped to in the last 2 years, we moved to the very big area. This is implemented study, and we started in 2021, and then we cover about 500 kilometers square. This is not small area. We covered into two districts, Bantul and Sleman. The population is 1.5 million. This is a big population. So just wait. Looking to the map, I think it's clear up. If you probably you already familiar with Yogyakarta. So the city of Yogyakarta is just small area. The red and the red border in the middle of the map. And then when we talk about small scale release, we're talking in the four very tiny area, for tiny black things here. The size is around 0.2 or 0.7 kilometers square. So we selected 4 because this is actually to assess how can the Wolbachia establish in this area. So this is small thing, and then we move to the impact area in the middle one, which is 32, and then now we're working in the big area. So from the mountain, here is the north area is the mountain, and south area is the beach. So you can see the challenge not only about the community, but the challenge is also about the environment, for the higher level of the altitude and the lower level of the altitude. And then if you also listen before, if you also follow the previous presentation, Ari, and then also the previous one said that the temperature is the main concern. So this is also the challenge in us in Yogyakarta. So we start with a small scale release. So this is a very simple thing actually, just to demonstrate the feasibility, the establishing Wolbachia. Because before is the story is about in Australia, and Ari involved that. So everything about the Wolbachia story is in Australia, and then we bring it actually in the story to Indonesia. This is just simple thing to bring it, but the very complex one, because this is new approach. As you know, that's already there were existing strategy, existing control method that is contradicted with what we want to try. First is about talking about the population rejection, population control, but no, it's actually in the releasing mosquito. So that's why I would mention about the community engagement is very important. So as I repeat again that this is just small area. And then this is isolated, and Ari involved in this process. And then select from 10 area, and that's you survey. We have a criteria. We monitor the population there. So what we're doing before we release the mosquito in this area is really rigid, really complex. So first is actually we need to see how is the population of Wolbachia there. So how is the community acceptance level in there? So we try to select the most feasible area, because this is a trial thing. So after everything is ready, everything is on place, regulations already got, community already prepared, and then and also we prepared the mosquito, because a lot of question is actually about the sensitivity of the mosquito that we release it in the field. So people, our stakeholder and also community concerned about that we suspected that we released the Australian mosquito. So that's why we did the back crossing before. So we pushed the mosquito to mate with a Wolbachia mosquito to mate with a local mosquito, Aedes aegypti mosquito, in a several times, just to reduce the Australian background, the mosquito Australian background, and then move it to the Indonesian background. So we did we did 6 times. And then this is actually we released in the community. Release, we also experienced with adopt release. If we hear the before question, we experienced with adopt release as well. And then we doing the weekly. And then 40 adults release in a 25 meter square. And then ticket for 20 rounds. Every week we release the adult, and then for 20 rounds. But the problem with this adults, actually we release the very hungry mosquito. We keep it the mosquito in the lab, and then when we release it's actually in the position of the empty of the stomach. So when we open the cup, the mosquito hunt the community, and we got a lot of concern about the mission, about the mosquito population, about the mosquito biting. So that's why from this actually we try to step back and then look what is actually the most possible listen to the community, and then we change it from the adult to the egg release. So in the egg release, we're doing the weekly with a container. Also, we adapted actually in the technology with actually that applied in Australia, but we totally we changed in some way to just adapt the environment in Indonesia, and in terms of the design, in terms of the hole and everything. But this is just kind of the container with a 1 liter water, and put it eggs there, and then put it set up in the environment for 2 weeks, and then every 2 weeks we change it the new eggs, we give a food, and we get water, and then we did it actually for 12 rounds. So what actually we monitoring for the first, because this is really important step to go to the next level. So first is actually concern about safety. A lot of people actually asking about the similarity of genetic of the mosquito, not only the mosquito but also the Wolbachia in the mosquitoes. We did an experiment in the lab for genetic similarity. Actually, we collect the mosquito from the field, and then we doing the satellite microsatellite analysis. And then we comparing between the genetic of the mosquito with Wolbachia and the genetic of the white mosquito. And we got a really similar. Also, the important thing, as Ari said, is insecticide resistant. We're really strict on the insecticide resistant. The product of the back crossing, we tested many times to make sure that the insecticide resistant between the mosquito that we release actually in the same level with the mosquito in the white band, because this is about the mating. If we release the mosquito and then the community spread with an intensive insecticide, and then our mosquito actually has the low level of the insecticide resistant, so our mosquito will kill it in the higher level rather than in a white band. So fitness also, just because this is about a mosquito that we have expectation that they can mate with the white one to continue the Wolbachia to the next generation. So we check it the hatching rate. So hatching rate, we just count the number of eggs, specific number of it, put it on the water, and then count it on the 3 days. How much is actually in the larvae still alive there? So we expected actually in the mosquito that we released it to the field have the hatching rates more than 90%. And then of course is actually Wolbachia frequency from the material that we release should be 100%, because we don't want to release the vector. And then dengue, Zika, and chikungunya infection on the colony that we release it. We still using the human blood feeding. This is my son, so already 12 years actually feeding the mosquito. Still alive right now, except the getting skinnier year to year. I mean, you know, so the important is actually we have to make sure the community, they safe. They really feeling safe that mosquito, that Riris is actually the local mosquito, having the the not alien mosquito or not different mosquito in the fitness, in the insecticide resistant. And then the most important is actually that mosquito is actually safe from dengue, Zika, and chikungunya. In the field, we also, and the field is also important in monitoring. Of course, the Wolbachia progress weekly. It's really strict at that time. Actually, a lot of we're using the PG trap in really dense areas, about 100 or something. So in the one RT about 40 to 60 hosts. We set up at the 3 to 5 BG, and Ari actually really concerned about the distribution and support the trends of the BG in there. So because this is also the first, so we have to monitor carefully. So we collect the mosquito weekly, and then bring to the lab, identify, and then send the IDS-HFT to the diagnostic, and then screen for the Wolbachia. And then we're doing the after 1 or 2 years the establishment, we've seen that the Wolbachia is quite stable. So we change it from the BG to the Ophitrap sampling. So I mean, you know, Ophitraps in the bucket, and then we put it inside of the house with the water, with a flannel, and then the female mosquito lay eggs there, and then we collect the mosquito. Collect the eggs, hatch it on the lab, and then we screen from the adult, and then we check how much actually Wolbachia in there frequency. And also the concern is about the safety aspect, because this is the first. So we also tested the Wolbachia in human, the infection of the Wolbachia in human by withdrawing the blood from the community, also in the blood feeder, and then also we look at the horizontal transmission to check how is actually the Wolbachia safe from the environment. Is there any, you know, transmission for Wolbachia to other insect in the field? And also we check it also on the Wolbachia similarity, because again that the concern is you release the Wolbachia on the mosquito that actually relate before in Australia, not in Indonesia. So that's why we try what is actually in the concern from the community, question from community. We doing the monitoring, and then we doing the trial, and just to give the evidence, because actually already a lot of evidence on the paper, but here in the community didn't 100% believe on the paper. So they have to show by themselves. For example, the human blood feeding. This is also quite important actually to community to have the proven evidence that Wolbachia is safe, something like that. So the result, this is a quite long period. I think it's a 2014 to 2016. And then we got a result that, okay, the IDS-HFT infected Wolbachia was identical. So it means that we release the local mosquito with Wolbachia. The key is also important is insecticide resistant and fitness. And then we are about the dove that Wolbachia cannot establish in the high temperature in the tropical area. We answer here that Wolbachia really love Indonesia environment. We can check its ladder, and then Wolbachia is also safe for environment and human. This material for the first release is actually very important too, as the evidence to communicate it, the step attended. So I mean, this is actually the important thing. So it just give an illustration. This is already also on the Buchori paper. So it's how smooth actually Wolbachia progress in Indonesia. So the blue one is during the release. So during the release, Wolbachia improve. We stop when Wolbachia is already reached 60, and then they develop by themselves, and then it's quite consistent, stable. So 2014, and then we doing the monitoring again in 2020 and 2021. And then Wolbachia in this area is still really beautiful. I mean, the Wolbachia still over 90%. So it means that not only established, but established in very long period. It's more than 7 years. So this is, if a lot of people have a question about how long actually Wolbachia established in the field. So far in Indonesia, 7 years. 7 years is still okay. And then in the lab, we already maintain for, I think it's close to 100 generation, about 12 years, and then we never get a drop of the Wolbachia in the laboratorium. So this is also the key to answer the doubt about how effective, how long actually the Wolbachia keep the benefit or keep the impact on the reduction of the dengue. The second is impact study. So this is about the Kathy work, mostly about Kathy, and then the EPI team work. So in the study of the Wolbachia to reduce dengue. So first is we're talking about the establishment, mostly talking about the mosquito and the Wolbachia and the environment. And now we're talking about Wolbachia, mosquito, environment, and dengue. So how to evaluate how is the impact of the Wolbachia in the dengue case. So in the area I already mentioned. So we divided into two. I think it's a Uut already mentioned. We have a two design, quasi and a randomized control trial in the 32 kilometers square. But the method is, I think, is we move from the adult, which is really what you call it complicated in terms of the what you call it the community concern. So we moved to the eggs release. So we started in 2011, and then in quasi in 2016 and 2017 in the RCT. And then I think it's already mentioned about the acceptance. We use the public acceptance. So we're using the egg release monitoring still, but it's not really tied to before because we move from the safety to the real of the study. So we in the lab, we still monitor the mosquito fitness, Wolbachia frequency, dengue, Zika, chikungunya infection, and insecticide resistant, of course. And then in the field, because this is a study and we have a protocol. So we also monitor about the release quality. So we look at the bucket, and every single bucket we check it the status. This is fail or okay. Fail means that the Wolbachia has only a few of the pupa skin, and then we also monitor how much is actually mosquito that we release per bucket. So it's mean also to predict how much actually mosquito that we release to the environment. So also of course Wolbachia frequency. We still using the weekly PG trap, because this is research in the really straight vertical distance and the density. So in most is actually in the distance between one PG to PG is a 250 meter. So and then another thing is actually environment impact. I think it's we also monitor about how is the changing of the fogging, but in entomology side actually we look at how much is actually in the changing of the population, the population dynamic of the IDSHFT, also the resistant character, insecticide resistant character. So we collect the mosquito from the field back to the lab, and then we test it from the ER and then see what is the difference between the Wolbachia with in every, from the control area and also from the intervention area. So we check it, we compare it, and then we seen that we got a stable establishment like the first. First is in the small, it's really stable, but we learn from the bigger areas, about 30 kilometer area. We got also stable establishment in the area, and then there is no change of the population resistance. So your population and the resistance is not really changed between the Wolbachia with Wolbachia, IDSHFT with Wolbachia and IDSHFT without Wolbachia. And then the paper is already submit, still in the submission. So just to see, I just pick up two areas and the quasi and then Ahmad, how beautiful actually in the move of the Wolbachia. The green one is actually during the release. We put it about 13 to 15 times, and then we stop it when the Wolbachia is over 60%, and then let them to grow, to establish, and then we can see, for example, in Bener and Wirobrajan, this is a kelurahan in the quasi. Look at that. After stop the release, the Wolbachia is just so continue in the really high frequency. Also in the RCT and the randomized control trial, I just pick up two from 12 cluster, and then also the same with the quasi one, the Wolbachia is very stable one. And then this is the impact of Wolbachia and the population dynamic insecticide resistant. This is still under the submission, and we can see that during the release there is the increase of the Wolbachia population, but this is about 10 to 15%. But after that, there is no significant of the population dynamic between the in the control area and the intervention area. Also in the Wolbachia insecticide resistant on the mosquito, we can see that pre-release is same, that during releases between the Wolbachia, and then the treated area is the same as well. And then post-release is the same, and then overall is actually there is no difference between mosquito with Wolbachia and then mosquito without Wolbachia. And then pre-release actually this is the mosquito that what's gonna maintain the lab compared with the wild, but and the post-release actually in the bird, the mosquito actually collected from the field. The last actually implemented study.
Pak Warsito, you have like 1 or 2 minutes left, yeah?
Okay, thank you. Yeah, and this is not really far anyway. So this is the implemented study. I think this is more light because we already got the community already, you know, already got information about the impact of the because this from the second release actually we proved it that Wolbachia effective to reduce the dengue. So it's quite easier actually to communicate to the community because the community has a problem with dengue and they have hope that this kind of the innovation actually can help them to come up from the problem. So we're doing the release 2021 in Sleman and in Bantul is 2022. Monitoring is just about the lab is the same. This is but the monitoring in the fields is just the Wolbachia monitoring and then release quality. We don't concern about the safety anymore, the environment, because this is already answered in the previous what got released. So the results is community I think is really good in Yogyakarta. They really accept, and then the release actually not done by us but done by the community. As I would mention that the release actually mostly what is done by the cadre. And then this is just how to see how we monitor the quality in the release. So we check it the fail, the rate of the fail. So every week come to the buckets, just not all of the bucket we monitor, but only 10% of them. And then we check it, we expect it actually in the fail less than 20%. And then this is the number of the mosquito per bucket. So we expect it actually to get 140 per week. But here we can reach about 170. And from this kind of the good quality, we can see the progress of Wolbachia in Sleman and Bantul. Right now in Sleman and Bantul, the Wolbachia is already over the 70%. So we expect it to get 80% and then give the benefit like the FCT. So this is the last one for the overview. So we try to analyze based on the on the data that we collected the latest one. So in the Yogyakarta city, and the monitoring is 2020, Sleman 2022, and in Bantul release is still ongoing. But we monitoring it last month, and then we can see that Wolbachia already cover the whole area of the targeted area. The blue means that Wolbachia over 80%. The green is means that Wolbachia over the 60%. So we can see that Wolbachia really love the Indonesian environment, not like in Brazil that up and down, and then in other areas like Ari mentioned about uncertainty. But here in Yogyakarta, like this is a kind of that Wolbachia is really stable in the establishment in Yogyakarta. It's just a bit picture how is actually in the community doing the release using this is the bucket, put it X there. A lot of because the ownership of the community is really high. When they give a sign, they give a kind of fancy place to just to keep it the safety of the bucket. And there's a lot of discussion between Autumn with the community just to make sure that the quality of release is like what we expected. So that's all.
Okay, thank you so much, Bapak Warsito. So the next speaker we have Bapak Dr. Riris Andono Ahmad. I think he is ready here, and if we can read his short CV, that will be great. Okay. Okay, if it's not there, okay, there we go. So let me introduce Bapak Dr. Riris Andono Ahmad. He is the director of the Center for Tropical Medicine from KMK UGM. There's an English title about that, but I don't remember all of it. Maybe Pak Riris later can explain the English term for that. And he is also the co-principal investigator at the WMP Yogyakarta and a member of the COVID-19 response team with expertise on epidemiology and infectious disease control. So without further ado, Dr. Riris Andono Ahmad.
Thank you very much, Prof. Dami. Can you hear me?
Yes, we can hear you very well.
Thank you very much. Good afternoon, everyone. It's been a pleasure for me to share our studies with all of you. And I would like to share my presentation. Can you see this?
Yes.
Okay. So I will talk about the public health impact of the Bulgaria implementation, and I will cover these issues. One is the reducing dengue incidence, hospitalization, disruption of dengue focal transmission, impact on vector control activities, and also dengue case fatality rate. As previously mentioned by Prof. Adi Utarini, Prof. Uut, this is a long study with several phases, and I will focus on the phase 3 where we did the impact study. And Prof. Uut also mentioned that we have two studies. One is the quasi-experimental, and the other one is the RCT studies. And I will focus the study on the RCTs. Here is the study settings. These two on the left and the right is this quasi-experimental study. And on the in the middle, this is where we did the CRCT study, the OAT study. And this study conducted in around 26 kilometers square of Yogyakarta, whereby we have around 37 kilometer squares. We divided the area into 24 contiguous clusters, and we did a constraint randomization. Prof. Adi already also explained how we did the randomization. The study started from October 2016 when we did the community engagement, and then we did the randomization in January 2017. The release was started in March 2017. And then we start the clinic enrollment in January 2018. And due to a COVID pandemic, we had to finish the enrollment in March 2020. And the primary outcome is try to see the efficacy of this technology in reducing virologically confirmed dengue case incidence. Well, there are secondary outcome, but we will focus on the first primary outcome. And we intended to have 400 VCD cases, which can detect 50% reduction and 80% power. This is, Pak Warsito already mentioned how we did the intervention with the X-release and everything. And this is how we did the enrollment of the dengue suspected patient. We enrolled in 18 Puskesmas health centers in Jogja, which cover all the clusters. We have 53,000 patients visited the clinics, and we enrolled around 8,144. And of those, we divided into intervention, those from the intervention arm, around 3,700, and from the untreated arms for around 4,400. And we found 384 virologically confirmed dengue and 5,900 test negative controls. So this is already explained that we have efficacy around 70%, 77%, and the preventing the hospitalization around 86%. And I would like to also present this, which we compare with the existing dengue vaccine. We can see that the Wolbachia intervention somewhat comparable or even better with the dengue vaccine which is available currently. Especially if we want to see in the dengue virus type 2 and also type 4, and also in terms of hospitalization. So this is a good news. We also, this is already explained by Prof. Uut, that it's somewhat reduced the dengue incidence in Jogja, but I would like to also add on, because if we compare with the dengue situation in neighboring district, this is Yogyakarta, this is the study area, Yogyakarta, and this is Bantul district, which now we start to deploy in those areas. And we try to see, compare the dengue incidence, the trend of dengue incidence prior to the study. We can see that Bantul has somewhat lower and less fluctuative dengue incidence with a peak around 20 per 100,000. And in Yogyakarta, it's more fluctuative with the higher incidence. And during the study, we can see that somewhat the dengue incidence in the city reduced, while in Bantul it's somewhat similar. And after the post of the study, we can see that even the dengue incidence, the trend of dengue incidence in city, Yogyakarta city, somewhat lower compared to the Bantul district. Bantul district is less urban area compared to Yogyakarta. And if we see this is three different graphics. The first one is the distribution of dengue confirmed cases in the study. We can see that the dots, the different color of the dots represent the serotype of the dengue. The gray one is the intervention area, and the white one is the untreated arm. We can see that we have more case incidence in the untreated area compared to the treated area. And if we compare this is the fraction of test positive in the study. We can see also that in the intervention area, most of the cluster were less than 0.05. Only in the cluster 10 that we have somewhat in the between 0.05 to 0.1. And in the untreated area, we have a higher fraction of test positive. In this graph, we can see this is cases which have similar serotype. There are 163 enrolled serotype dengue with a homotypic pairs, which has a proximity of 300 meters from each other, and within 30 days of the onset of the illness. This is a proxy of local transmission. And if you can see also here, there are more these cases in the untreated area compared to the treated area. So this graph shows that the intervention can prevent a local transmission. Another part is this about the insecticide fogging. Insecticide fogging is very popular in the city. Most of the time before the study, we can see that we have a very high, similar between the control area and the in the intervention area, in the treated area. And it's comparable, the frequency of the fogging events. But if we compare after the intervention, we can see that in the untreated area, the fogging is still high, while in the intervention area is somewhat very low, and it's also correlates with the VCD cases. And even the good thing is the awareness of the community when we talk about fogging, they have a better awareness because there was a story during the study period. We have in the duration of political campaign, most of the political candidates they often give fogging for community to attract them to vote for them, but community in Yogyakarta city at that time said that if you want to give a fogging for us, you need to somewhat coordinate with the district health office because they are the one who decide when and where to do the fogging. So this is a good proxy that we have a better awareness in the community. And it also reflects in this factor control practices in the community because one of the resentment from many stakeholders is that Wolbachia technology will somewhat change the behavior of vector control. Particularly in Indonesia, we have this 3M, mengubur, menutup, and then menguras. So it's cover the water container, drain the water container, and bury all the disposal. They think that if we have Wolbachia intervention, this will lead to a reduction of those practices in the community. But our data shows that between the intervention and control area, there were no difference between these activities because this is where the cleaning of water container, where the community do regularly once a week. We can see that there is no difference between different activities, including covering the water container and also buried unused good for which can be a breeding site. It's also comparable when we talk about Jumantic. Jumantic is the vector control activities, vector monitoring activities. And this is part of a strategy from the dengue control program in Indonesia. We can see that over time, if we compare to these two different arms, there is no significant difference between the practice in the community. And lastly, I think I will talk about the dengue, the impact of the Wolbachia on the case fatality rate. We can see that due to the reduction of dengue cases, the reduction, we saw an increase in case fatality rate because we have a reduction of a dengue incident as the denominator. We still need to see whether this is somewhat accidental or whether it's a result of reducing awareness of community in the severity of dengue because they saw less dengue cases in Yogyakarta. So with this result, I close this presentation and I give time back to you, Prof. Dami.
Thank you, Pak Riris. A very, very amazing work and very wonderful presentation. Thank you so much. Okay, the next speaker is Kat Edenborough, and we will see a record of her. So allow me to read. So Kat Edenborough is a virologist. with over 8 years of experience. Her research has led to the development of animal model systems to facilitate viral and antiviral research. She is a researcher at Monash University and investigating dengue virus evolution and selection of Wolbachia-infected mosquitoes. She completed her study at the University of Melbourne with Bachelor of Science in Honors, and then proceed with a PhD on influenza infection. And now and she's been invited to numerous conferences, etc. So I think let us now listen to her record.
My name's Kat Edenborough. I'm a postdoc at the Institute of Vector-Borne Disease, which is situated at Monash University in Melbourne, Australia. And today my presentation will focus on evolution and implications for the Wolbachia approach. So far in this conference, we've heard many presentations that have highlighted the success of Wolbachia as a replacement method in curbing dengue cases. But personally, I think one of the big unknowns with replacement methods is the durability of protection that Wolbachia affords. So durability of protection is likely to be influenced by the evolutionary stability of the genomes of each organism within this tripartite interaction. So the dengue virus genome, the Wolbachia genome, and the Aedes aegypti genome. Today, I'll be speaking to the evolutionary stability of the 3 separate organisms. And then I'll really focus on dengue virus evolution and the risk of this occurring to Wolbachia. So the Wolbachia genome. The genome of the W-mal strain of Wolbachia, which is the field strain used for releases, is 1.3 megabases. It contains several prophage sequences, a lot of repetitive DNA sequences, and mobile genetic elements. The stability of the W-mal genome in the field has been investigated in several studies now, but I'll speak to one of these, which was conducted by a PhD student, Kim Dainty, at our institute. So what Kim did is she collected short-term and long-term mosquito samples in cans following W-mal releases, and she sequenced these W-mal genomes in these samples. And what she found was in her short-term collections in 2011, she identified 2 SNPs. One was an error in one, so SNP is a single nucleotide polymorphism, so it's essentially a point mutation in a nucleotide within the genome. So SNP A was an error in the reference genome, and SNP B she identified in 3 of 10 samples. And then in her long-term sampling, she identified an additional 4 SNPs, but these only occurred quite rarely. So they're only in 1 or 2 of the 30 total samples that she collected. But again, she found SNP B, which seemed to be a little bit more prevalent. So I think what this indicates, is if we look at the size of the genome, the detection of only, 6 SNPs within this period of time is actually indicating that there's generally a low level of diversity and that the genome is quite stable following introgression. And what Kim also found is that this trend is reproducible across different sites. So far, Kim has sequenced W-mal genomes in Indonesia, Vietnam, and Colombia, and she's also finding only a very small number of SNPs in a small number of mosquito samples. Okay, so the Aedes aegypti genome. So it has 3 chromosomes, it's 1.25 gigabases. It has a lot of repetitive sequences, and a large component of it is made up of transposable elements. I think there isn't really sufficient information to make any firm conclusions about the stability of the genome in the context of Wolbachia infection. So I think it's quite, questionable. But, just from what we've seen in the field, that this WMAL-induced dengue virus blocking and also mosquito fitness phenotypes seem to be very stable in the field up to more than 10 years. We're seeing only small number of breakthrough infections in cans, despite a number of travel infections coming into Queensland every year. And I think there's been only one study that really directly looks at the stability of the Egypti genome in following release. And what they did is that this is from Ari Hoffman's group, where they sequenced Aedes aegypti genomes pre and post released from 4 towns around Cairns. And this study identified more than 100 SNPs that were associated with WMAL infection. But what they also found is that geography had a greater influence on nucleotide diversity than WMAL infection did itself. So I think this really highlights some of the complexities of these studies, where geographical differences might confound signals of SNP selection and make it difficult to kind of link SNPs that are associated with the presence of Wolbachia. So the dengue virus genome, it's a small genome, 11 kilobases. It's encoded by a positive sense non-segmented RNA. And I definitely wouldn't describe the dengue virus genome as stable. So we know that there are 4 serotypes. Each serotype has emerged independently from the salvitic cycle. And these have diverged into several genotypes, which we can see here in this phylogenetic tree for each serotype. And so, the different serotypes and different genotypes, they can all co-circulate in the same region, and this is known as hyperendemicity. And serotype and genotype replacement events are associated with increased dengue cases and severe disease outcomes. So it's kind of key to the epidemiology of dengue. So the phylogenetic dynamics of dengue is this rapid lineage turnover. And that's where you kind of get a shift in the population genotypes that are present in a given year, away from the dominant genotype to kind of a subdominant lineage at an earlier time point. And this lineage turnover really does contribute to virus diversity as well as the multiple genotypes. So I think given this diversity, dengue virus evolution is really an obvious risk to the potency of the Wolbachia approach. And so it's something that's really worth further investigation. So how we're investigating this risk, dengue virus evolution and the resistance risk, is both in the lab and in the field, and then trying to use our lab studies to inform our field data. So in the lab, we aim to identify sites of the dengue virus genome that's subject to WMAL selection, and for any variants that we identify, to characterise their fitness and resistance profiles. In the field, we're aiming to establish a framework for dengue virus surveillance in Wolbachia release sites, and using that framework, conduct surveillance for resistant dengue variants. And then we'd like to merge these two together and explore field sequences for our experimentally determined resistant dengue variants. Okay, so I'll speak to the lab studies now. So a PhD student of Cairns, Kian Dong, she's based at Oxford University Clinical Research Unit in Ho Chi Minh City. She optimised a dengue virus serial passage system in WMAL infected mosquitoes. So essentially, what Kian did is she recruited febrile dengue 1 positive patients, so 6 independent patients, into a study, and she blood-fed mosquitoes with these dengue positive patient samples, and she fed them to both wild type and WMAL mosquitoes. And then after feeding them, she left them for 10 days, collected the mosquito homogenates, and then re-injected those homogenates into the cognate mosquito line. And she did this for 20 passages, so trying to adapt the virus to the different mosquito lines. And so here, just to let you know, we had 3 separate cohorts for each patient for each mosquito line, so 18 mosquito cohorts for each line, and each mosquito cohort was made up of 20 individual mosquitoes. And then to really monitor virus evolution over passage, what Kian did is she sequenced the virus population. So she did this with Illumina whole genome sequencing. And then she developed a kind of filtering criteria to pick out the SNPs that were likely to be under positive selection. So she looked for SNPs that were induced a de novo mutation, a non-synonymous substitution, so they caused an amino acid change in the consensus sequence and were at high frequency. She looked for SNPs that increase in frequency temporally and over passage. She also focused on SNPs that were reproducible, that they occurred in different patients in parallel. And so today I'll really only speak to the SNPs that met all of these selection criteria and were likely to be positively selected. So she identified 2 sites in the dengue genome under selection. So these codons were both within the envelope protein. And so the envelope protein of dengue virus is a structural protein, and it's on the lipid membrane of the virus particle. And just to let you know its function, so it mediates attachment and entry to host cells. So what she found is that for one of these positively selected SNPs or codons, that it was only really occurring in W-mal mosquito cohorts. And this was this glutamic acid to lysine mutation at residue 203. So it was quite prevalent. She found it in 7 of 18 mosquito cohorts from material that originated from 4 independent patients. And we didn't find it to be selected in any of the wild type cohorts. And so we think the function of this mutation might be involved in fusion thresholds. And this is because, it's a glutamic acid to lysine change. It's a change in charge. And if you actually look at the position of residue 203 on the 3-dimensional dimer of the envelope protein, we think that a change in charge at this position, might impact how the envelope protein actually undergoes rearrangement, and that might impact pH fusion threshold. So the other positively selected codon that Kian identified was at residue 384, and this was selected in both mosquito lines. So we found it, in several cohorts in both wild type mosquitoes and WML mosquito cohorts in several patients. And so this mutation is situated in an attachment loop in domain 3 of E, and we think that the mutation might increase the affinity for mosquito receptors and could have resulted from just the conditions of the passage system, like differences in temperature, or only requiring to infect mosquito cells and non-human cells. So what we were then interested in was the stability of this E203K mutation. So we performed an additional 10 passages for 3 different cohorts of the passage 20 material. And we chose cohorts that had a high frequency, so greater than 50% frequency of this E203K mutation to determine the stability of the variant. So when we passaged these E203K high virus populations in wild-type mosquitoes, we found that after 10 passages in all 3 cohorts, there was a really rapid decline in its frequency to less than 10%. And this mutation was actually replaced by the progenitor sequence. So that was interesting, and it looked like it wasn't, either very fit, or it was either outcompeted by the progenitor sequence here. But when we passage the exact same material, so the same cohorts in WML mosquitoes, we found that this mutation, the E203K mutation, actually became fixed. So it was, reaching above 95% frequency here. So I think this really is a key experiment, and it highlights the dependency of this E230K change and the selection of this on the dependency of selection on the presence of WML. So to summarise our lab evolution experiments, we've shown that if we passage dengue 1 in WML infected mosquitoes, we see selection of codons in envelope, and this is an E. Specifically, we see this E203K change, which is really reproducible. When we try to characterise these variants, it appears that they're negatively selected in wild-type mosquitoes, and we've also looked at their we've passaged them in human cells as well, and we find that the frequency of 203K also declines and is replaced by the progenitor sequence. These mutations aren't prevalent in the field, so they're in less than 5% of E sequences that are deposited to sequencing repositories like GenBank. And so we think that these type of variants might not survive the human transmission cycle if we know that they're kind of unfit in one host. We find that they're not very fit in human cells, and we're not really detecting them in circulating sequences. Founded on our lab results, we think it's really important to perform surveillance for dengue virus in sites of WML establishment. And with the support of the WMP team in Yogyakarta, we've been able to start a field study for virus evolution. And it's been done as an ancillary study to the randomised controlled trial that was conducted in 2018 to 2020. So far we've sequenced 37 dengue virus whole genomes that were collected from patients recruited pre-released in 2016 to 2017. And 326 genomes for post-establishment RCT samples collected between 2018 and 2020. And together the pre-release and post-establishment samples will form our baseline sample set. And don't forget that the majority of the samples here the cases here are occurring in untreated clusters. So what we then plan to do is sequence dengue genomes in 2024 to 25 from any virologically confirmed dengue cases in this time window as part of a post-establishment monitoring protocol. And then we'll compare baseline and prospective sequences to identify changes in genotype or signals of selection. It's likely to be challenging to study virus evolution following WML introgression because there's likely to be few dengue cases in sites of WML establishment. Any cases are likely to be travel infections and not linked via local transmission, which means it's quite difficult to detect any signals of selection. But anyway, I'll take you through our analysis of baseline samples to begin with. All right, so what we did here is we assigned sequences for each virologically confirmed dengue case to a genotype with phylogenetic trees. And what we found is that, most genotypes persisted. They were present in pre-release and post-establishment. But there was only one additional genotype, dengue 4 genotype 1, that was observed post-establishment. And what's most obvious here is that dengue 2 and dengue 4 had higher prevalence than dengue 1 and 3. And this is in contrast to the previous season where dengue 1 and 3 were the most dominant serotypes. So this might suggest that a serotype replacement event was occurring in Jogja at this time. So we've gone back and looked at the trial results again, which were initially calculated on a serotype level and published as a serotype level, but we've since integrated genotype into our intention to treat analysis. So when the number of virologically confirmed dengue cases in intervention versus untreated clusters are compared on a genotypic level, and so this is really only relevant for dengue 1 and dengue 4 that had multiple genotypes, the mean efficacy is really very similar to the serotypic level efficacy. And this suggests that the protection in Jogja is covering multiple genotypes within the serotypes. What we also did is define lineages in the post-establishment samples. And this lineage designation was based on genetic distance and similarity to sequences deposited in GenBank with a known country of isolation. So after we assigned lineages, we determined that if they were locally acquired, so if the lineage contained a pre-release Yogyakarta sequence, or did not, otherwise they were designated as an imported sequence. And so through this analysis, we identified 15 lineages local to Yogyakarta and 14 lineages that may have been imported from elsewhere in Indonesia or elsewhere within the Asia Pacific. And so we expect that the 14 imported lineages are likely to be an overestimation, as we didn't sample as extensively in the pre-release period. So, but it still demonstrates that the lineages in Jogja, there are many of them, they are quite diverse and dynamic. And then what we did is we looked for each lineage, we looked for the number of times that individual countries shared lineages with Yogyakarta, and this was just based on phylogenetic relatedness. And then we made a network. And so what we can observe here is that the countries shown in orange, they seem to have stronger associations to Yogyakarta, so they share more lineages than other countries shown in yellow or blue. And so these include Taiwan, China, Singapore, and of course other areas within Indonesia. And finally, this is my last result that I'm showing, that there's no evidence of experimentally determined codons, so our E203K and E384K mutations that we observed in our lab experiments within our Yogyakarta dengue 1 sequences. And if we just look at substitution rates on a per serotype level, the number of substitutions per site of the viral genome per year is really consistent with previously published values at around 6 × 10⁻⁴ substitutions per site per year. So it doesn't indicate that evolution for these viral genomes has been sped up. So to summarise the field evolution, I've shown genotype level efficacy for the trial results. I've showed that there's diverse lineages in Yogyakarta. There seems to be a biasing towards lineage sharing between Jogja and specific countries within the Asia Pacific. And we couldn't find out experimentally determined variants in our Yogyakarta sequences. Okay, so I'd like to acknowledge everyone in this slide. So thank you all my lovely collaborators for helping me with my research. And I'd also like to thank the organising committee for giving me this opportunity to present my work. Thanks.
Okay, thank you so much. So we have listened to all the 3 speakers, very wonderful presentation, very exciting, all new information. And now we have about, 26 minutes for our discussion. And I see that there are already people raising hands and also questions. So I see this one person who has been raising hand right from the first. So I would like to give the opportunity to Pak Faiz Mahfudz, because he's been raising his hand for more than 30 minutes. So I think I should give him the opportunity to ask by himself. Pak Faiz, tadi mau raise hand, ada yang mau ditanyakan atau tadi kepencet? Okay, he's gone. So I guess he's not raising his hand anymore. Pak Faiz, are you there? No. Okay. So I'll just start reading the question. And okay. Oh, okay. Pak Faiz said, I'm sorry, it was an accident. Okay. So let me ask the question. I'm going to read the first question. Actually, there are 3 questions from one person. So allow me to read it, and then Pak Warsito, Pak Riris, and also Kat, if you can position yourself on which is most appropriate for you to answer. Because I think the question is more basic than the monitoring and the impact study. So, okay. First of all, Mosquito, also the Wolbachia strain inside, that is now established in Yogyakarta and originates from Australia. and the mosquito, okay, can survive in Yogyakarta. Okay, he wants to be corrected if he is wrong. I'm sorry, there's no name here, so I don't know who's questioning this. The first question is related to the sex of the mosquito is released. What is the sex of mosquito that is being released? Is it only male, female? So maybe that's Pak Warsito, yeah. The second question is, did you also research whether there is any different Wolbachia survey in wild mosquito in Yogyakarta? So maybe if there is any Wolbachia strain from the wild mosquito, will there be a bidirectional mating? Or if no Wolbachia and a mosquito, will there be unidirectional mating? Okay, so maybe this is also related to Pak Warsito. If the males and females are released together, does the mosquito bring Wolbachia to invade the population in Yogyakarta? And the third is, how does the ecological affect the release of your mosquito to the wild population? Will the original mosquito from Yogyakarta extinct? Okay, so this is more about the ecology of the Wolbachia. So I think maybe these three questions can go to Pak Warsito first.
Thank you, Budami. Thank you, the person who has the question there. So first is about the mosquito that we related to the ecosystem in Yogyakarta is actually, yes, the first is actually originated from Australia, but then we not directly released to the field habitat, to the open habitat or to the wild habitat, but we did the back crossing when the mosquito with Wolbachia with the Wolbachia mosquito with Wolbachia with the local one. So this is actually the mosquito that we released to the field is actually the mosquito that already breeded in the laboratory between the Australian background and Indonesia background, and we already detected in the end of the product of the back crossing that the mosquito is identical with the local one. So it's not really Australian background or Australian mosquito with the Wolbachia, but the local mosquito with the Wolbachia. So the first question is about, yes, we release a female and male so far. And then about the second question, we did a survey of the mosquito Wolbachia, but not specific on the mosquito. But we also explored the Wolbachia that infected the other insect, including the butterfly and etc. And we found that more than 50% of the wild insect have the Wolbachia, but specific one the Aedes aegypti so far we didn't do survey, because a lot of paper actually mentioned that Wolbachia not present on the Aedes aegypti. So for the infection, actually infection not about the mosquito Wolbachia invade the wild population, but the Wolbachia actually invade to the wild population. We're not replacing the wild population with the mosquito that we produce in the lab, but the concept is actually we release the mosquito and that they're mating with the local one, and then continue the Wolbachia to the next generation. So, but still also in terms of the infection, we never get 100% of the wild population of the mosquito having Wolbachia. So it's usually close over the 90, but we never reach 100% in the wild population. And then ecological aspect, I think is temperature is not really matter in Yogyakarta, probably because in the same area. So far, we can release in the really close to the pitch, which is quite warmer than in the highlight. But the most factor that influence the progress of the Wolbachia is actually kind of physical boundaries inside of the ecosystem, because the mosquito actually move has the limited movement. So this is a domestic mosquito. So if one host and the distance of one host far to the next host, so it takes time actually to mosquito to move from the host that we put it the bucket to the host that far from this location. On the city, for example, because the host is actually crowded, there is no space between the one host and another host. It's quite faster than in Bantul, where the one host is actually isolated from another host. But again, that yeah, rainfall probably, but it's again that it's not really a big challenge. But we still, I mean, you know, mostly it's about if rainfall coming, I think it's they trigger the hatching of the the deposited eggs that probably most of them actually the eggs that no having no Wolbachia. So sometimes Wolbachia a little bit drop, but then if you continue to release, they will go up again and then reach the establishment. So far that.
Thank you so much, Pak Warsito. I'm not sure, is Kat here with us, or is she? I know that she has COVID, so maybe the organizer. No, okay, so Kat is not here. So we have Pak Riris and Pak Warsito for question and answer session. Okay, Pak Riris, there's one thing that really amazed me from your presentation, and that is the impact. From what I see, that reduced fogging. It's just, you know, as an entomologist and working with pesticide, for me, this is like, wow, this is such a huge impact on the reduced fogging itself. So I think one of the questions I would like to ask you is, I know this RCT is just in a certain area, but are you also going to do a spillover test, for instance? Are you also going to look at whether this Wolbachia infected Aedes aegypti can exist in other areas that has not been released? And are you going to check also the fogging situation in the area, or how are you going to pursue this forward?
Thank you, Ramami. It's interesting questions. Yes, this is the data from our study, but when we did the release in Sleman, we haven't really got the data, but according to their program manager, dengue control program manager, they also saw a reduction of fogging after the release of Wolbachia. They said that before they implemented Wolbachia, I did not remember exactly the number. They have, I think, 3 times higher budget for fogging. And after the Wolbachia release, they see also a reduction in budget, but also it's not yet all spent after the release. So I think it has a similar effect in other cities other than Jogja. And we also expect that will be similar when we finish the release in Bantul. And I hope this is really happen in areas in the future when we have this technology implemented.
Okay, thank you. Sorry if I want to ask one more question, because I'm just amazed with the data about the vaccine. You also showed that it's comparable between the dengue release and the vaccine. And for me, this is such a game changer. I mean, you're changing the pesticide uses, you're changing the vaccine. This is, I mean, we talk about impact. This is what we mean by impact. But do you see a resistance? And well, you know, people are already used to. I mean, how should I say that is quite sensitive? Do you think there will be any resistance from the people who have usually used fogging, etc., and then also vaccine? You know, I mean, this is a business of vaccine and a business of pesticide, and this is a game changer for everyone. Have you thought about this? I mean, yeah.
Well, I think again that we what you call put this product as a complementary strategies rather than competitive. So if we can have different strategies. And I think it's now common that in every disease now we use what we call after the COVID a Swiss cheese strategy, that each strategy will reduce a certain risk, but will not reduce all risks. So if we can have several complementary strategies, it will ensure that we can control and most possibly eliminate dengue in the future.
Okay, wonderful. Thank you so much. Well, there's another question here that I would like to read to you, and this is related to the WML. Why WML is injected in Aedes aegypti can survive and block the dengue virus. So he or she is asking the genetic mechanism and asking about what about the WML alien strain in Aedes aegypti. Okay, why not choose Wolbachia strain that originates? Okay. So I don't know, would Pak Warsito answer this or Pak Riris?
It should be Ari or Cams to answer about that.
Yeah, I thought it should be Ari. But it should be Ari.
But I mean, you know, we working not only and in early stage actually we not only working on WML. You know, in Yogyakarta, even though I mean, you know, we working with a WML pop, and then also another strain WML pop, which is, you know, really has the good blocking capacity, but suffering mosquito. So we predicted actually, and that if we release this kind of the strain, probably we got a problem with establishment. But also we maintain with the double infection of the WL, albu B, and something in the lab. But it's really, really complicated in the production in the lab. So that's why we back to the WML, who has really good in the blocking, but in the really good in the what you call association, in the stability association between the Wolbachia and the mosquito. And so far, it's proven also in the field that the progress of the infection is really smooth, and then the long establishment actually recorded in all area in Yogyakarta.
Okay.
Yeah, we really, you know, headache with previously we really headache with another strain actually. Yeah.
Could I add to the answer?
Yes, please do.
Thanks. I think it's worth remembering that WML is from Drosophila melanogaster, and it's a globally distributed fruit fly. And if you look at the genome of WML that we've used in these Aedes aegypti mosquitoes, the genome of that WML is the same as the genome of WML in Indonesian fruit flies. So really, it's don't think of this as an Australian WML. This is an international WML, and it persists in Aedes aegypti because it sets up a stable infection in the germline. So the ovaries of the female mosquito that ensures transmission of Wolbachia from one generation to another. So it's a very stable infection once it's in Aedes aegypti.
So you're basically saying that WML does not have nationality, right? It's just the same.
It's a globalist. It's international.
And also we check it here as well, Budami, and then we get a evidence that WML that on the Drosophila here is actually identical with the WML that infected on the Aedes aegypti. So this is a clear evidence.
Okay, clear evidence. Thank you. Thank you, Cameron. Okay, the next is, this is a question to Pak Warsito, because according to the question here, he's asking about impact. Yeah, how to technically assess the impact, because he's not really quite clear from your presentation. According to the speakers, sort of missing the point. Okay, Pak Warsito.
Yeah, I think it's Pak Doni will because this is about epidemiological monitoring, but I can see that not only monitoring, we not only monitor about the Wolbachia frequency, but also working with the Dinas Kesehatan, Bantul and Sleman. We also monitor the case of the case of the dengue. And then so far, I mean, it's quite early actually to conclude that how far actually will give it the impact, because this is we just finished the release in Sleman, and then also we still ongoing on Bantul. But it seemed I met what you call it, Dinkas Sleman last few days, and then about the presentation, they said that there is kind of the promising evidence that after the post release of the post establishment of Wolbachia in this year 2022, in the case of the dengue dropped, they recorded about 200. That compared to the Bantul is about more than 800 cases during this year. So this kind of it's kind of a good sign, a good clue that Wolbachia is quite working well in Sleman as well.
Thank you, Pak Warsito.
Pak Doni,
Pak Doni, would you like to
monitoring of the dengue case impact, etc.
The main impact will be if we can interrupt the transmission of dengue in the community, and we will have a study, upcoming studies, which try to see whether after the intervention, we will see an elimination of dengue transmission in the city. So let's see. I mean, there will be an upcoming studies try to see whether we can provide evidence that there will be an interruption of transmission of dengue in Yogyakarta city.
Okay, thank you, Pak Doni. Okay. Next is a question that is related to the situation of the COVID-19. Since the work is very laborious, how did you keep the project going during the pandemic situation? So I think both of you should answer this one.
Yeah.
Pak Warsito.
Pak Doni. Oh yeah,
Pak Doni first.
Okay. There are two levels. At the more at the organizational level, we try to minimize our contacts within our teams itself. But then we still, because of, especially in Sleman, we even have to stop the release because of the PPKM. It was level 4, so we cannot do anything. But I think Pak Warsito will describe how he really carefully handled the activities in the fields, so that we can still follow the regulation. Please, Pak Warsito.
Yeah, the good thing from this technology is actually don't need to contact with the people. So you just set up the bucket. So you can come to, and then without knocking the door, without meet someone, after you get approval from the community, and then we not kind of asking the people to crowd in somewhere in the activity. So I think it's in terms of the Sleman and Bantul, I think it's we really follow the protocol. We just give it. We try to minimize as much as possible the contact with even the community, and then the cadre, and then if the kind of capacity building, we can doing through the WhatsApp, doing the call, or even we produce a lot of video just to give the overview how actually we handling the technological operational thing on the field. So and then we send it to the cadre. So it's actually the things that why on this technology can be still operationally on the real, even in the high incident of the COVID one. But in Bantul, actually there is some period that we stop it, because this is not because of the technology is not really applied on the kind of the condition, but the cadre actually doesn't have a brief to a lot of cadre actually infected by the corona, the COVID. So we cannot ask other people actually to replace the cadre, because it seems that we have to train them from the early to the thing. So yeah.
Okay, thank you. I think there's one more question that I would like to ask Pak Doni, and this is related to the future. That We've listened to Kat's presentation on evolution, and what she did was more at the laboratory level, and very interesting result. And if you take that into the field, it would be interesting to see the evolution of you know, the virus evolution and also the resistance risk. And since the project itself is already finished, how are you going to deal with this? Is there going to be still some studies that is continuing just for the impact and the monitoring study, or what's the future for this?
Currently, we are in discussion, and I think we almost start the upcoming study for assessing the impact of this technology, particularly whether it can eliminate dengue in Yogyakarta city. But I think it's an open venue for other studies relating to dengue evolution, whether it will change how the circulation of dengue virus in Jogja or other things. So it's an open invitation even.
Okay. So from an epidemiologist point of view, if someone wants to come, let's say people from Undip, they want to come to Jogja and to conduct a study on epidemiology here in Yogyakarta, that is something that is possible to do. Yeah, I mean, it's open for everyone, right?
Yeah. I mean, it's Yogyakarta city. It's not belong to UGM.
Okay, wonderful. Since
but the point is actually we have a lot of treasure.
Exactly.
You know, we sampled the mosquito from the early until now. We keep it on the biobanking. We still have on the field. So this is kind of the open opportunity to explore. And then I think it's a camps already, what your camp and our team has already done for a little bit when you try to see the mutation, etc. But still, you know, in the next, a lot of things to explore in terms of the ecological, in terms of, you know, genetic things, and in terms of the community even.
That's a very good point, Pak Warsito. I think what you guys have done here is really the frontiers, and I mean Ari and his team infected the Aedes with Wolbachia, and then you guys took this and use it, implementing, and then now you have policy. So this is really from science to policy. And then I heard now that the government is also embracing this technology. So this is a real success from that point of view. And I hope that it will continue the research, because research never finished. And there are still many interesting questions, many interesting questions and things to pursue. Just like Pak Warsito said, you have a whole treasure of information, and I hope the collaboration between everyone in Indonesia and also abroad can still continue. So maybe just one closing statement from both of you, from Pak Warsito and Pak Doni, if there's just one closing statement that you would like to say. We still have about 2 minutes.
Thank you very much. Our study shows that the technology can somewhat interrupt the transmission. We expect that it will eliminate dengue in Yogyakarta city, and we hope that we can see the similar result in other cities in Indonesia in the future. Thank you very much.
Thank you, Pak Doni. Pak Warsito.
Yeah, I forgot to acknowledge all people who work very hard, you know, from the first to the end, you know, from our lab, from the what you call it in the field, and also the community, and then what you call it, the scientists as well, camps, and the ARI and whoever, you know. It's not really easy to reach in that point. And the most important, actually, thank you very much for Tahia, who really patient, you know, support us in a kind of uncertain condition at the early, but, you know, I think it's a quite proud together that we are, you know, what you arrive in that point, which is not only scientific but also in the community level. Yeah.
Thank you. Thank you so much.
Thank you, Bu Dami, as well.
Big applause
thank you.
to
You
Pak Dami.
invited here, Prof. Dami.
Yeah, thank you.
Pak Warsito. Thank you so much.
Ngopi-ngopi, Bu.
Ngopi-ngopi, yes. So I'm giving back the time to the moderator. Thank you.